Welcome back, everyone. Today I wanted to talk about an interesting paper that was published last February, February 2024, in the journal Paleoentomology, called Unusual Snakefly Larvae in About 100 Million Old Amber and the Evolution of the Larva Pupa Transition. And if you have been watching these videos, this is from the same lead author that did that mystery, that did the paper on the mystery larva that was found in that Cretaceous amber. This is found in similar Cretaceous amber, but it's about snakeflies. So the next obvious question is, what's a snakefly? It's something that most of you probably haven't ever heard of. And this is a snakefly. They're a very weird group of insects. They're actually their own order called the Rhaphidioptera, related to the Neuroptera, the lacewing flies and stuff like that. And if we go to the cladogram, where we would find them is way down towards the bottom. They are right here, the Rhaphidioptera, the snakeflies. So they're related to like the lacewings and things like that. And they are one of the early holometabolous insects. So one of the first insects to develop an actual metamorphosis where they go through a pupal stage into the adult stage. And those are the snakeflies, named as such because their larvae and kind of their adults have this weird long body. So, what do we know about the snakeflies and what does it have to do with this paper? So this is what a snakefly larva looks like. All snakeflies, as far as I know, are predatory, and we do have them here in the United States, although you'd really have to be looking for them. They tend to be associated with trees or soil or hide under bark, and they tend to be very slow growing and frequently pretty small. So you'd really have to be looking for them. Frequently, they can stay in this larval stage for a couple years, and they might not get more than a centimeter in length. Some of the times they can be very small. There are a few large ones, but frequently they're very small. This is what the larva looks like, and you can kind of see where it gets its name, being the snakefly. So they go through a metamorphosis. This is their pupal state. They look very different, but the Rhaphidioptera, the snakeflies, have something weird about their pupal state in that their legs are not bound to their body, and they are actually still capable frequently of walking around. So they have an active pupal state. They're not helpless like what you would think of a butterfly cocoon or chrysalis. They're not trapped there. They can actually move around quite a bit. And there are some major changes between the larval stage and through the pupa into the adult stage. Some of those changes, other than the wings, can be missed by someone who's never looked at a snakefly before. So if we zoom in on the snakefly and compare it to the larva, other than the wings and the color differences, there are major changes in the head. You have the formation of the eyes, the mouth parts enlarge and move forward. The head kind of gets narrower, it gets drawn in and pulled forward. The pronotum here becomes elongated compared to what it is in the larval stage. And then there is a general elongation and then condensation of the pterothorax into this muscular structure. But compare all that to this larval stage, the head is flatter. If you look at it from the top, it's kind of wide. The pronotum is generally either equal size to the head or shorter. What will become the pterothorax, the meso and metanotum, are undifferentiated. They're not very long. They're actually pretty short. And then you have this kind of undifferentiated the undifferentiated abdomen. So a lot of changes occur in the pupal stage. So when they get into the pupa, they molt this exuvium or their final larval skin, and they begin immediate development of the head. They elongate the pronotum. They start developing the wings. They get rid of the stomata, which are these larval eyes, and they replace it with these giant compound eyes. So there's a lot of changes that go through this one stage that we call the pupa. So now we get into the point of this paper. I've included some drawings here just to make it a little easier to see. These are the stomata, the larval eyes that get lost in order to develop into the adult compound eye. The mouth parts are smaller. You can see that there's not a ton of differentiation between the abdominal segments. They're all kind of the same length. In the larva, the pronotum here is either equal or shorter than the head, and that can be differentiated with the adult structures here in this drawing, where you have the much more obvious ocelli, the little eyes on top, the large compound eye, this kind of narrowing of the head and the pronotum, and then obviously the development of the wings. So let's get into the research. What they found in this Burmese amber is this Rhaphidioptera, the snakefly larva. It's about 100 million years old. And what they noticed by looking at it is that it had some really weird features. And the features don't line up with what we consider the modern snakefly anatomy. So things that you might notice compared to the larvae the line drawing and the pictures that we just saw, is that the pronotum is already really narrow and long. The other two thoracic segments, the mesonotum and the metanotum, or the mesothorax and the mesothorax and the metathorax, are already beginning to differentiate. They've become narrow. They're not the same size and shape as each other. Like when we look here, they're kind of all the same, except for that smaller prothorax. They've begun elongating, they've begun differentiating, and it's still in the larval state. There's clearly no wing development. The head is still kind of the larval head here, but the mouth parts are beginning to move forward. They're getting longer. They have moved away from the head, similar to how the adult mouth parts are. And then there's also some changes in the antennae and the tarsi, which are a little harder to see in this fossil. So this was a little weird. This seems to be as though the larva is almost going through a pre-pupal metamorphosis. They haven't actually moved into the pupal stage yet, but they are already exhibiting a mixture of developmental processes associated with the pupa and also associated with the adult. This characteristic of exhibiting mixed developmental stages within one life stage is called metathetely, and it's something that is known to occur within snakeflies, but it's usually artificial. You have to rear the snakeflies under very specific laboratory conditions in order for this to be induced. And it's not something that you would normally see in nature. And it's to see this in an amber fossil would be very strange because it's considered so rare and kind of artificially, has to be artificially humanly induced. So it raises some questions about the early holometabolous insects and how they developed the ability to pupate. So one of the things that the researchers did was that they wanted to see how weird was this this thoracic elongation compared to other snakefly larvae. And so what you have here are the triangles are extinct species, and this is the head to prothorax ratio. In modern larvae, they should be about the same length, or the prothorax should be shorter. And then the squares are pupa and larvae that currently exist. And if you see where the squares are kind of bunched, you can see that things that currently exist are very different from this particular species. This species really has nothing directly around it. So it's not a morphology that is currently extant or even known from the fossil record. So this is definitely something weird is going on. So like I said, this raises questions about holometabolous insects and the evolution of metamorphosis. So when we think about extant metamorphosis, or how insects currently do metamorphosis, and the holometabolous insect life cycle, that should be 4. I can count, I promise. We go from egg to larva. The larva eats and eats and eats and eventually gets big enough that it triggers pupation. The pupation is one life stage, and then the adult emerges from it. That's the butterfly life cycle. This is the ladybug life cycle. And it's the same for beetles, even though beetles and ladybugs aren't particularly closely related. And actually, this process is the same for all modern holometabolous insects. All insects that go through a metamorphosis go through a single pupal stage, and they follow this same pattern: egg, larva, pupa, adult. However, something that many of you that don't work with all different kinds of insects might not know, and if you don't work with any insects, you really wouldn't know about this. There are insects that aren't holometabolous that kind of walk the line and actually have like two pseudopupal stages, and these are thrips. So this is the group Thysanoptera, and if we go to the cladogram, I'll show you where they are. They're up here, and they ride this line. So holometaboly is developed here. So this is hemimetaboly, and this is holometaboly. So no metamorphosis technically, and a true metamorphosis down here. Thysanoptera, or the thrips, are right on this line. And they actually do kind of go through a metamorphosis in that they have their nymphal stages, and then they go through a stage where they kind of start to develop wings, but it's not a true metamorphosis. But what's weird about them is that they tend to have two pupal stages, these sort of pseudo-pupal stages. And then the adult emerges from these pseudopupae. And again, they kind of run on this line, and there are a few hemipterans that do this as well. So you can kind of see the development of pupation and holometaboly in these two groups here. But what about the groups down here? Do we see any evidence of that? And this amber fossil, or this amber specimen, provides evidence that actually there is some sort of development of adult and pupal characteristics occurring in these early holometabolous insects in a larval stage, almost like a pre-pupal stage. So this was the conclusion that they were drawing from us. The purpose of this paper is this is what we think of when we think of modern snakeflies. It goes from larva and it molts directly into the mobile pupa. And this, in the pupal stage, is when you start seeing the adult characteristics, which is what you see here. You clearly see adult characteristics in this one pupal stage. But what they are proposing is that in the early snakeflies, they were still developing the process of pupation. And so it wasn't a distinct stage. Pupation could actually be divided into two stages. The larva would molt once and develop a modified thorax and head, and they would start developing the adult characteristics. Then they would molt again into their pupal stage, which would have definitive adult characteristics in the head and thorax, and then the wings would develop into what we consider their normal pupal stage, and the abdomen would begin to develop into what we consider the normal pupal stage. So at least sometime in the Cretaceous, this was still occurring in the holometabolous insects, even though today we don't see this at all. So this is actually a cool fossil that allows us to see into the gradual development and evolution of what we now take for granted as this complete metamorphosis insect life cycle. So that's really, really cool. This paper is open source and or open access, and I will link it in the description if you want to read it. It's a very short paper because it's basically just a description of what they found and then them trying to explain what they found. So it's only about 8 pages, very fast to read if you want to read it. And I definitely encourage you to. A lot of the papers from the Journal of Paleoentomology are free. So I'll link that and I'll talk to you guys later. Bye.